Hello, good evening, good afternoon to everyone here. Thank you for joining me for this really fascinating discussion with regards to Robert Malone. Now, it's important for people to realize that actually I had the first video interview with him all the way in June 2021. And at that time, it was just purely by chance that I realized subsequently that he was actually the inventor, the creator of the mRNA technology. That in itself was a fascinating insight for me. And since then, I have watched him grow from strength to strength, going across the world and effectively impacting the scientific perspective and across the world. Now, today is actually my birthday, and the biggest gift that I've had is the fact that we have here Robert Malone with me to discuss ideas about mRNA technology, his history, how he felt about the Nobel Prize, and all these issues together should, in truth, be a remarkable discussion. So, the first thing that I'm going to do is bring Robert in. So, for us to just have a quick introduction. Hi, Robert, how are you?
Hi, Philip. Thanks a lot for having me back. It's been a long time and a lot of water under the bridge. And
Absolutely.
I'm grateful for you asking me to come back onto your broadcast.
Well, yes, as I said, it's a tremendous honor for me to actually have you because these days you're literally one of the biggest voices out there. Depending on which side of the fence you're on, it can be a bit of a problem, but in truth, everybody does hear what you say, whether they like it or not. So, with that in view, can you just give us an introduction of yourself, Robert? Let us know a little bit more about you and your background.
So, the truth of the matter is that I actually live and enjoy living a simple life. I live on a 50-acre horse farm in Northern Virginia that I homesteaded together with my wife, Dr. Jill Malone, from just being hayfields. I was born in Palo Alto, California, at the old Stanford Hospital. Grew up in the Central Coast of California, moving frequently. The son of a Mills University trained or college trained teacher and a Stanford trained electrical engineer who was very involved in the defense industry and high voltage systems. I landed in Goleta in my youth and pretty much grew up in the Santa Barbara metropolitan area, and then went to university at Santa Barbara City College, UC Davis, Northwestern University, UC San Diego in the SALK Institute, and I think I mentioned Northwestern and Harvard Medical School for my final fellowship in global clinical research. I'm trained in pathology. I've also in virology, immunology, molecular biology, and was very active as a bench researcher, really beginning about 83, 84, kind of cut my teeth with HIV and mouse mammary tumor virus, which is a retrovirus model of breast cancer in mice. And then through my work in gene transfer, first at the Salk Institute, focusing on retroviral vectors, then as a assistant professor at UC Davis, I grew a medium-sized non-viral gene therapy program, was recruited to University of Maryland, Baltimore, where I had additional discoveries that led to a whole nother field of gene delivery that nobody talks about these days, having to do with pulsed electrical fields, and the founding of a company called Inovio that's still running in San Diego using that technology. And then to the Uniformed Services University of the Health Sciences, where I helped create a breast cancer research program and tissue banking laboratory and research laboratory up in Western Pennsylvania. And then joined a company that had just obtained what's called the Prime Systems contract for biodefense products for the Department of Defense, called Dyneport Vaccine Company, up in Frederick, Maryland, almost across the street from Fort Detrick, which is where the United States Army Medical Research Group is, that used to be the biowarfare group, but now is the biodefense group. And at that, in that job, in that position, I had experience with virtually all the major, well, really, and minor vaccines that are in development for biodefense for the U.S. Army and defense complex, which is where I was brought into connection with a lot of people that are called, referred to as deep state. Many people who have CIA connections. I'm not CIA. I've never been CIA. I've never been approached by the CIA, to the best of my knowledge. But you can't work in biodefense and not work with CIA people. I've also worked for a Gates-funded nonprofit, one of the first, called ARIS Global TB Vaccine Foundation. It's defunct now, but I know that world pretty well. I've consulted for PATH, which is one of the core Gates companies. And I've worked for a for-profit regulatory affairs sweatshop in Rockville, supporting FDA-related work. I've written many INDs, presented to the FDA multiple times, written many clinical protocols, and managed phase 1 and 2 and some phase 3 protocols, either as a principal investigator or as a support person, like a clinical investigator or safety committee member. I've was at the tip of the spear in bringing forward the vaccine candidate from Newlink, originated from Public Health Agency Canada, and getting it sold to a company called Merck. That's now the only licensed Ebola candidate, or Ebola vaccine, and have been very involved in advancing many different biodefense products. I've kind of been at the forefront of, I think, 5 different outbreaks. Did a lot of drug repurposing work with Zika, after I determined that building a Zika vaccine was not going to be very practicable. And many influenza vaccines. I have deep experience in influenza and influenza vaccine development. I'm very familiar with the Novavax platform, and worked for a long time for a company that uses baculovirus to express proteins in caterpillars. And I've won over my lifetime well over $10 billion in grants and contracts for a variety of companies, including companies that are very much beltway bandits, basically inside DC defense contractors. I've been granted secret clearance. But my passion remains in small farming, and my 44-year marriage with my wife, Dr. Jill Malone. We're high school sweethearts. And these days we produce Lusitano horses here in our farm in Virginia, up against the Shenandoah Mountains. So there's a kind of a broad brushstroke of who I am and what I do.
Excellent. Listen, I think as I listened to you there, I remember similarly the first time that I had an interview with Gert, and he went through his background, and I thought, oh my goodness, no wonder it is a worry when you say something that is counter to the narrative. And we're going to be coming into that in a moment. But I think it's very important to start with that question of mRNA technology. Now, in all of the background that you have listed, I don't hear a lot about the work, or that original work with mRNA. Why did you not continue after you had that experience of the breakthrough in thinking? So you would have been at that cutting edge, as you said, in Salk Institute. You had that moment where you envisioned it. Why did it not continue?
Well, not only envisioned it, but I left the SALK because of the politics that were going on over the patents. With a diagnosis of post-traumatic stress disorder. My mentor is now outed basically as being a very abusive person. But I continued that work at a small startup across the street called Vical. And it's at Vical that we had the additional discovery of naked DNA and RNA delivery into mouse muscle and the reduction to practice of all of this for the purpose of vaccination. So we demonstrated there that you could inoculate mice with the messenger RNA that I had synthesized and purified and cause them to mount an immune response against the encoded protein.
And so is that where the patents come from? Because
Yeah,
looking
there was
behind you,
there
there
was
are
an original patent filed. So the original patent disclosure was made at the SALK in 1987 or 88. And then a patent was filed from the SALK, and I never got a straight answer. When I left the SALK, it was still pending. They had first determined that I was a sole inventor, and then they put my mentor on it. And then I left and went to Vical. And I asked the SALK a few years later what happened to that patent, because they're supposed to notify the inventors of any actions they take under the Bayh-Dole Act in the United States. And they had unilaterally decided to abandon the patent at a time when my mentor, Inderverma, had been a consultant to the company Vical. So the SALK patent would have interfered with the Vical patents, and it was dropped. And then the Vical patents, there was 9 of them that came out of the original filings that were kind of 1990, 1991 that issued. And then we had, Jill and I had a subsequent patent, actually multiple patents, that's as shown behind me. Those are just some of them. And they cover the formulations, cationic lipids, the use of various cationic lipids. We were the first ones to put cholesterol into the formulations. We came up with solutions to the endotoxin problem, et cetera, et cetera. But we were way ahead of our time. And Vical, when I left, I had signed an employment agreement that, among other things, resulted in my getting compensated for the patents with $1. But I also signed away any rights to work in that field subsequently. And at one point, I got a cease and desist letter from the CEO of Vical, B.J. Samant, who used to be at Merck, that I should not do any research as an academic directly relating to what I had done when I was at Vical. So Vical was very aggressive about the intellectual property. They sold it to Merck. Merck tried to make DNA vaccines work. They thought RNA wasn't going to be feasible to manufacture. They failed. They spent over $2 billion. They turned the IP back to Vical. Vical burned about $1 billion or $1.5 billion, and they failed. But what they did do through that whole time is very aggressively block anybody that would work on DNA or RNA vaccines until the patents expired. And then after the patents expired, the market cap for Vical basically crashed, and the company was basically merged for less than cash value.
So is that where you then start to speak about your work? Because before that you couldn't.
I really couldn't. Yeah, well, no. At that point, in all this work that we did mostly at UC Davis with the different positively charged fats and the pharmacology and biology and mechanism of action work, we had done a lot of work on trying to develop gene therapy for cystic fibrosis involving pulmonary or intratracheal inoculation of these formulations in mice and in monkeys, in non-human primates. And also the basics of delivery, including jet injection and pulsed electrical field. We were starting with some in gold particle delivery, which is another technology in this mix that has gone by the wayside. And with the positively charged fat formulations, we could never overcome the toxicity. They're highly, highly inflammatory. And so we moved on to other delivery technologies that were more efficient, including pulsed electrical fields that didn't have the toxicity. and primarily focused on the DNA delivery, not the RNA, although we did get a very broad patent on mucosal vaccines that covered virtually any gene therapy vector.
So, mhm.
But I couldn't get any of that funded.
Yeah.
I got recruited to Maryland, and we did the electroporation work, and I wrote what I think was probably one of my best personal proposals ever. And it went into the NIH for review, and it was rejected based on the fact, quote unquote, that Merck had already solved the delivery problem, which of course they hadn't. But this is the way things are. And when you're a soft money young investigator, you don't have too many times at bat. I guess cricket would have to be the metaphor for you. For me, it's baseball. But if you don't hit it out of the park as a soft money investigator, you're out.
Yeah.
You have absolutely no security. And I had a wife and a child, 2 children at that point in time, and I couldn't tolerate that the environment of modern academe for a soft money funded investigator. And this is why I went to industry again and then to government contracting.
So let me ask you then. So from a personal point of view, you had to almost walk away from your baby at the time.
Actually, I did, and it's actually a little even more abrupt than that. This is not my first rodeo in being a whistleblower. I was one of the lead individuals speaking to the press about what I knew concerning the death of Jesse Gelsinger at the hands of Jim Wilson and UPenn from overdosing, going off protocol with adenovirus vectors. And I knew what happened there was I was taking training in bioethics from a prominent bioethicist at Maryland, as was my wife. And I went to him and said, this has happened, and I know these things about this. They're not being discussed. They're not available in the press. And he told me he thought I had a moral obligation to go to the press and share what I knew. And so I did it knowing that it would probably destroy my academic career, and it did. But I was able to recover, survive. I got the appointment at the Uniformed Services University of Health Sciences, and was able to kind of rebuild a whole new career. But I made a conscious choice there to speak out about things that I thought were unethical, and had to live with the consequences. But what I learned from that was that if you maintain good friendships and good relations with colleagues, you can survive a blow like that. They'll help you. I use the metaphor of a rock and roller throwing himself onto a mosh pit. You know the image that I'm talking about, and finding that people would catch me and hold me and allow me to carry on. And with that comes a huge sense of security that you can act in accordance with your morals, even though it will often have major consequences, and still survive. But the thing about it is that you come through that and you still own your soul, which I think was worth it.
So here is an important piece of the puzzle that I'm trying to unveil, Phil, and it's to do with the fact that you would not have been able to get through that period without the support of Jill. And secondly, I know that Jill was a primary factor in you speaking out about your work with mRNA, because you could have kept silent. You could have just let it ride. Why did Jill insist that, listen, you need to share this. You need to talk about this. Why was she so sure about that?
So remember that when I left the SALK, I left with a diagnosis of PTSD. And that's not a trivial thing. I don't just say that. I was a hot mess. And that's something that doesn't go away. I continued throughout much of my professional life to find myself in highly competitive academic environments where the same kinds of behaviors are common. And I had also, since those early discoveries, really had a heck of a lot of gaslighting and ridicule and from people who benefited by positioning themselves as more important than they were in the story, and building companies off of that, etc. And so I had learned a survival strategy of basically staying quiet and laying low, being not readily visible. I certainly would speak at conferences, but didn't make much of. I basically set aside that early trauma and the events associated with it, and just went on with my life and built other skill sets and other research focus. And so when this started to heat up, and frankly, particularly, I was triggered by a comedian in Canada who posts videos under the pseudonym What's Her Face, that did a video that really went through and dissected what happened to my Wikipedia page after I was on the Dark Horse podcast and all the wicked editing that happened with that to write me out of history. And at the time, there was a clear marketing campaign to get Carrico and Weissman the Nobel. This is 2.5 years ago now. And my wife, who has lived through all of this, experienced it firsthand, Dr. Jill Malone, said, "No, Robert, this time I insist you have to stand up and fight what's being done to you." And I really, I'd partitioned all of that in my mind and set it aside, and really didn't look at it, didn't try not to think about it as a defense mechanism. And she hauled out. We'd carried the old data to the extent that we were able to extract it from Vical and the SALK. We'd carried it with us in a great big box through all the moves and academic appointments and everything. And she hauled it out and went through it and pulled out key documents and assembled them and wrote an essay in which she described, from her point of view, what had transpired, and included the receipts, the documents, and the patent disclosures, etc., and sent that off to UC San Diego faculty and leadership, SALK Institute, people at Moderna, an old colleague that had invited us to the Karolinska years and years ago, that was involved in RNA delivery and became a collaborator and was on the Nobel Selection Committee a couple of years ago, and basically said, no, this is what's happened. So that kind of set that all in motion, the process of objecting to being written out of history, and having my own contributions overlooked. And of course, there was massive backlash from corporate media, notably a very ugly piece, poorly written, in the Atlantic Monthly that has a graphic with two syringes crossing over my face like skull and crossbones, with my image in red. It's very striking. Somebody wrote the other day that it was basically an invitation for assassination as far as they were concerned. But I took a lot of arrows over that, and the Atlantic Monthly, the Washington Post, the New York Times, Business Insider, Forbes magazine, all of these entities, as well as my haters in academia, particularly at the Scripps Institute of Research. There's one that I don't want to amplify. they all focus just on the published manuscripts. None of them will acknowledge the patents.
Yeah.
But
So
because I
I,
was at a company, all of that stuff had to go into patents, not into papers. And so it's weaponized that, well, I didn't really do what I did because I don't have the peer-reviewed publications. I only have a few. They happen to be among the most highly cited in this area, but that's all whitewashed over as if that.
I want to understand, Robert. Now, this is an interesting question. As you were talking, I thought about it. Just imagine that you had stayed silent, as you had typically done. You said nothing, and what happened still happened. How would you have felt? Would you have felt the same, better, worse? What do you think?
I think it's good in it's been particularly therapeutic starting with your broadcast, but so many others where I've been asked to tell the story of what transpired at the SALK and at Vical. And there's a school of thought that when you have a traumatic event, you need to talk about it many times before you can heal from that event. And I'm a much better person, even though it's been rough. I'm certainly healthier psychologically because I have gone through this process of uh, sharing and expressing what I've experienced. And I've had feedback from so many people, particularly graduate students that have, and postdocs that have had to live in this really rather twisted modern academic environment, expressed to me, I so much appreciate what you said. I had similar experiences. These are what happened to me. And I think the whole thing, not to sound too touchy-feely, is been very therapeutic, not just for me, but for a whole cohort of people that have also experienced these types of rather avaricious behavior from high-profile academics.
I'm going to share something here now. So this is the Nobel Prize. This was the 2nd of October. So this is the PDF from the Nobel Institute. And they have here Catalina Carico and Drew Wiesman. I want you, because you knew this was coming, and I want to understand because I'm thinking about it from a scientific point of view. When you've done work and you've created ideas and then not to be credited for it, it does hurt. Did this hurt? Even though you knew it was coming,
So,
did it
so
hurt?
um, to reel back, when Steve Kirsch and I went on the Brett Weinstein Dark Horse podcast, the three of us sat around for about 20 minutes before we started the broadcast and discussed the potential consequences of what we were about to do. You could call it speaking against the narrative of safe and effective COVID-19 vaccines. And we thought at the time Steve was the one that was most bomb-proof because he had his own company. It caused him to lose his company. His board of directors basically asked for his resignation. Brett, certainly, he got demonetized. That was a huge impact for him. My understanding is he was quite depressed for a while. As you would well imagine, if that's your main source of income, getting demonetized from YouTube. And for me, the discussion was that the Nobel was in play at that time, and that this would jinx my ability to prevail in that or be included in it. And so it was a conscious decision that was a risk, consciously taken, but that it was the right thing to speak at that time. And so when this dropped, I was in one sense surprised, in another sense not surprised. I was surprised in that my colleague from the Karolinska that I mentioned, who's a full professor and works on RNA delivery and self-replicating RNAs, had assured me that the committee 2 years ago had reviewed the contributions of Carico and Weissman and had determined that they were not sufficiently meritorious to warrant the prize. And the committee at the time apparently was surprised because they had not been aware that I had done that original work. And as apparently Moderna and Pfizer were not aware because they didn't cite the patents. And that's another story right now. They're busy in a patent battle. And now Pfizer, paradoxically, is citing those old patents in their attacks against Merck. Against Moderna. So I had been told that the committee had reviewed this and found that it was not sufficiently meritorious. And my expectation was that the committee was just going to stay away from this topic, certainly for many years until it had declared itself, let's say, in terms of what were the real issues with this product, these mRNA vaccines for COVID and the technology platform, et cetera. But clearly they decided not to. They decided to take it up, and they voted to grant this in a very odd way. It is, and you've got it highlighted there. Basically, they did a carve out just for this to make this award. And the wording is crucial there, as is the press release that corresponds to this. The carve out is restricted to just mRNA vaccines against COVID-19. So this is a Nobel Award for enabling the rapid development of mRNA vaccines against COVID-19, not for the development of the core technology platform or anything else. So as Jill and I were pondering this and, how do we process this and deal with it? Her point immediately was, well, actually, this is kind of vindicating because the committee has not, has explicitly not made the award for the invention of the technology, which was what was being advanced in the press, in Stat News in Boston and others, CNN originally, before I started objecting, was that Carrico and Weissman were the ones that had created this technology. When in fact they didn't even start doing any of this stuff until 15 years after what I did. So in a way, it was okay. Now, the assertions that are made by the committee are largely false and fairly transparently false. That the assertion that Carrico and Weissman produced these 4 papers that involve the use of a pseudouridine modified base incorporated into synthetic RNA, reducing inflammatory responses relative to non-modified RNA. They hold the patent on that from UPenn. And that patent's fascinating because it says nothing about vaccines. And Carrico and Weissman originally did not intend to work on vaccines, but rather were focused on cancer when they did these discoveries. It's absolutely true, as the committee asserts, that Carrico and Weissman made these discoveries. And they made them after I had left, and I've congratulated them on that achievement of the Nobel Prize for their discovery. The next statement that's made there in that clause that justifies the product and in the press release is they talk about effective vaccines. That's highly debatable. And they also assert in the press release the vaccines have saved millions of lives and greatly reduced disease. That's also really in flux right now, that thesis. But you notice they speak of effective, they don't say safe and effective, which has become the mantra. So they dropped the safe part. And then in their press release, they talked about that what was really important here was that the pseudouridine invention was what was enabling for the rapid development of these products. And that's also a false statement. The discovery phase and development of the, let's say, composition of matter, that's technical patent talk, for a vaccine is usually, and in this case also, a very small fraction of the overall time necessary for development. The reason why these vaccines were developed so quickly had nothing to do with mRNA, but largely had to do with dropping the normal processes for assessing pharmacology, toxicology, biodistribution, pharmacokinetics, in phase 1, 2, 3 clinical trials, including dose escalation, and not to mention the non-clinical and clinical toxicology. So it was the dropping of the regulatory norms that resulted in this being done so quickly. It wasn't really anything to do with pseudouridine.
Well,
And
it raises a good point because I was asking about this as I was doing my research. I realized that actually they even modified the pseudouridine even further by creating an N1 methyl pseudouridine, which is what they've used in the vaccines, which is quite remarkable. I guess what they're saying is just the concept of using this pseudouridine is the innovation. It doesn't matter how you then subsequently
um,
adjust it.
yeah, that could be the logic. I'm not aware. In remember when I was at the SALK, I think there was 6 or 8 Nobel laureates there, including Francis Crick. I'm not aware in the history of the Nobel Prize for Medicine or Physiology that those that originated the technology were not included in the final award. That's very unusual. And the point that all of these, let's say, irregularities or untruths that are used to justify this award really, in my mind, speak of the logic underneath this not really being grounded in rigorous science and medicine. I think it has more to do with politics. That's a controversial statement, and I really don't want to come across as sour grapes or denying in any way or casting shade or disparaging the achievement of this or to second guess the Nobel Committee. They made their decision. But I do note that Pfizer is a major donor to the Karolinska Institute. So whatever happened here, it happened. I was surprised. You asked about my emotions. I was
Hmm.
surprised. What was more hurtful for me than the actual award was the rekindling of the bots, trolls, and social media scientists' attacks on me, asserting that this demonstrated that I didn't actually do what I did. That's just hurtful, and it's not based in fact, and it's tearing off the scab over the psychological pain. Frankly, I don't want to sound like a whiner of what was what was deployed against me by corporate media 2.5 years ago. But that's the modern ecosystem in social media is this kind of really rather wicked behavior of people that just want to hate.
Yeah.
And there's nothing to be done about it. You just kind of have to suck it up, snowflake, and deal with it.
Well,
And
it does raise.
that was the one that was more hurtful than the decision by the committee. I could have used the money, but Carrico and Weissman have already scooped up the Lasker Award, the similar award from the Israeli government, the similar award from the Spanish government. There was clearly a big push here, and this is big science, and that's just the way big science goes. And if you don't, one of the things early in my career at the SALK, one of the competitors to Inder Verma was Tony Hunter, who was also ran Nobel laureate. I guess I'm now an also ran. And for some of his key work, brilliant man, mind like a trap. And he once said to me, if you think this is the last major discovery you're ever going to make, then you should get out of science right now. This is basically buck it up, uh, you know, and don't whine. And this is just the way things are. Science is a hardball game. And so, and the other thing about this, you asking about the emotional state, my entire professional career, I've had to deal psychologically with the gaslighting and denialism and all of this. And so I've kind of almost had a lot of that emotional baggage burned out of me.
You know, I'm going to say something now, and I had responded to a few comments on social media as well. And as I wrote something, I reflected on it. And my feeling is that the technology has some limitations, but my honest instinct is that they did it with the wrong disease at too big a scale. Meaning
Absolutely.
that if they had done what the intention was, which was to focus on the high risk cohort and try to protect them with the technology, I think the risk or the potential exposure of the technology would have been significantly lower. And I said something here. Yeah, I'll finish with this statement. I said that no matter what the political situation is, the people who have been hurt in one way or the other by the technology will never be silent. And that's, in effect, the thing that is unstoppable. And so the platform, even though it could have more modifications, more benefits, could be destroyed at this altar. Any thoughts on that?
So I had warned about this, and not just the platform, but the entire vaccine enterprise has been compromised now. And public faith in public health, the reputation of the CDC, the reputation of the FDA. I speak about this from time to time. I was speaking to a Portuguese audience about a year and a half or 2 years ago, and an older woman asked me a question and prefaced it with a preamble. And she made the point that throughout Europe, the FDA had always been believed to be the gold standard for integrity and rigor in regulation of drugs and biologic products. And that she had come to now understand that the FDA was corrupt and could not be relied on. And when she said that, I had not allowed that thought to come into my mind up until that point. And it literally hit me like a brick to the head that in this rush, this Operation Warp Speed, and all of the propaganda and misinformation and disinformation that was propagated by the government, and the withholding of information about adverse events, for instance, myocarditis in particular. What we had done as a United States Health and Human Services and biodefense infrastructure and really biomedical pharmaceutical complex is we had destroyed or severely damaged our competitive position in the world as the preeminent developer and manufacturer of these types of products. And my prediction had been then, and I think we're seeing it accelerate, that what would be the logical consequence would be that the United States would continue to lose its dominant position in biopharmaceuticals to India and China, one of which is considered a hostile nation, and the other one is at best a frenemy, certainly a strong competitor. And that's what's happened. The United States now has to import not only final drug product, but drug precursors if they're going to do that. And the reputation of the FDA is trashed globally and within the states. The damage that's been done here through the logic of emergency use authorization, of products that have proven to be neither safe nor effective, in response to what was basically marketed or propagandized as a highly lethal respiratory virus with a 3.4% case fatality rate, based on Ferguson's modeling largely at Imperial College and his group, and is proven to be, paradoxically, Mr. Trump got this right. Early on, there's multiple video clips of him saying that the fatality rate here, or I think he calls it mortality rate, is well less than 1%. That turns out to be the case. And the whole logic that this was a highly lethal virus that would overwhelm our hospitals, cause us to need to have mass graves and freezer vans full of dead bodies, and et cetera, that's all turned out to be false propaganda. But that is what has been used to justify these breaches of what are really international norms of ethics and product development that have been developed over decades. And so we're now left, I could tell you my colleagues in the clinical development and earlier phase pharmaceutical development space are perplexed now. That it's what's happened in the States with regulatory affairs in the FDA is that what had previously been a fairly rigorous and highly structured process. We all knew the rules. We knew what the process was. We could project how much it costs. We could project the timelines and, grind away for a decade to get the product developed and, safe and effective. And now it's kind of anything goes if they want it to go. And if they want to hold you to higher standards, they will. And you can't predict what it's going to be. People are left with, well, who do I have to pay off? What do I have to do it's the whole system is now compromised. Its integrity is compromised. And it has all kinds of implications.
I want to ask you a part of the question, because another piece of the puzzle, even though we speak of mRNA, the delivery system with regards to lipid nanoparticles, and I heard you speaking about this recently, and I'd also had a recent conference, and someone, Christy Grace, had made some very important comments about this. And I just brought up here a paper that shows major delivery systems for these mRNA.
By the way, those graphics are grossly naive.
Oh, are they? Yes. So, all I want to know is that when you see the different strategies for delivering the mRNA, have we ever at this scale used the LNP in this kind of way? Are there any risks with that, separate and apart from the mRNA?
Yes is the answer, and we know this because of data that was shared by Moderna about their phase 1 dose escalation trials with their influenza vaccine candidate. It was shared to their stockholders. It wasn't made public, which shows that at the 100 mic level with the flu expression vectors, RNA, they had an enormously high adverse event rate. Something like 30 or 40% of the patients enrolled at 100 mics had developed serious adverse events over a fairly short period of time. So, because a lot of this work is not being done rigorously and by basically treaty, based on a meeting that was held, chaired by Margaret Liu at the World Health Organization a little over a year ago, the decision has been made within Western regulatory authorities that the cationic lipid formulation that you were just referring to, that, by the way, if somebody, in my opinion, if you were going to give the Nobel for the enabling technology rather than the platform, The people that should have got it was Peter Cullis at University of British Columbia and his colleagues, because that is the enabling technology, is that the lipids and formulation structure and the use of PEG that is used by all CureVac, Moderna, and BioNTech, Pfizer. That's who really made the breakthroughs after decades and decades of hard work. And I've known Peter Cullis throughout my career as one of the top lipid formulation specialists in the world. And if I can just beat this dead horse a little further, there were 3 companies in the running originally. The third one was called CureVac. And they were funded by an obscure investor that you've never heard of before called Elon Musk. And they ran a more conservative phase 1 trial with antibody titers as endpoints in humans, in which they used a significantly lower dose than even Pfizer did. Remember, Moderna is 3 times Pfizer, and CureVac was a fraction of the Pfizer dose, but CureVac did not use pseudouridine. And it got a robust antibody response, but not as high as those that had used the higher doses. And they couldn't get funding to carry forward with the product development, and it was dropped. But those data demonstrate that you don't have to have the pseudouridine. It works just fine without it. And Moderna didn't used to use the pseudouridine. They made a decision kind of at the last minute to license the pen patent. But the cationic lipid formulations are intrinsically toxic. The mRNA and its immunosuppressive characteristics, because this isn't really mRNA. This is a chemically synthesized molecule that has pseudouridine of whichever form throughout its backbone. That's not a normal characteristic of RNA. The placement of pseudouridine in normal RNA is very strategic and specific in where it's placed, not all the way through the molecule. And it is absolutely immunosuppressive. That was the intention, but that's a double-edged sword, especially when you're dealing with immune responses. So, and then there's the spike protein. And if we compare, for instance, by the FDA and CDC's own admission, the Novavax product, which is caterpillar cell produced and caterpillar virus produced spike protein, the adenoviral vectored products, that one of which, the J&J, had been sold in the United States until recently, and the 2 RNA products that also express the spike, they all have the common feature of cardiotoxicity, the myocarditis and pericarditis, and yet their core platforms, when used for other purposes, do not show that characteristic. So major problems with myocarditis and pericarditis and coagulopathy were not problems with prior adenovectored vaccine candidates. They haven't been problems with prior Novavax technology candidates. So I think it's reasonable to conclude, you're a scientist, that this is basic Sesame Street stuff. We have these different products. They have one common characteristic. They cause this spike protein to be present in the body at very high levels in a very abrupt kinetic, very different from the natural infection, and systemically rather than mucosally. So very different, again, from natural infection. And they all have this common feature of cardiotoxicity and coagulopathy and stroke and other things. And so my thesis is that we have a cluster of toxicities that are attributable to the formulation itself in the presence of a polynucleotide, because you can't make these particles, the Peter Cullis particles, in the absence of a polynucleotide. They have to have a polymer, an anionic polymer, to self-assemble, based on ionic interactions. So the lipid particle is toxic. The mRNA is immunosuppressive. That's a toxicity. And the spike protein is toxic. Those toxicities overlap, and the vector sum of those are particles and a process that is very complex from a pharmaceutical standpoint, and involves multiple active new chemical entities.
So
That's important from
I
a
want to
regulatory
ask
point.
I want to ask you a question, Robert, and I want you to think about from a scientific perspective as a scientist. I am baffled why the scientific community as a whole, the public seems to have clued in, a lot of the public, that something isn't quite right. Why is there this almost ideological
Yes.
perspective, which is unbelievable?
Yes.
How do you explain that?
So, okay, I'm going to do a little fallback. I serve as an expert witness on cases from time to time. And one of the things that's been drilled into my head is you can never know what's going on in somebody else's head.